Hello, and thank you for attending our webinar today. I'd like to introduce Dr. Namyong Kim, CEO of Curiox Biosystems, who will give a short presentation on the DA cell technology. We will then open it up for a Q&A session. Please type your questions in the chat box. Now, I am pleased to present Dr. Namyong Kim.
Thank you, Sheila. Thank you everyone for joining Curiox first webinar on the latest drop rate product, DA cell. My name is Namyong Kim. I'm a lead inventor of a DA cell technology that I am presenting today. DA cell product enables fast and precise wash and staining of suspension cells without centrifugation by taking advantage of the well-lessness of our DA plate. Interestingly, I would say that the DA cell product was the outcome of open collaboration with the scientists we met in the biopharma industry. We have known and worked together for over the years. Particularly, I'd like to thank three scientists I put down on the title slide. Dr. John Forbes from Amgen and Dr. Arunan Kalyaperumal at Eli Lilly, who was previously at Amgen, and Dr. Ilona Karif at Merck. I plan to make my presentation short today and instead spend more time on questions from you listening to this webinar. So as Sheila mentioned, please submit your questions in the chat box window whenever during the presentation, and I'll answer them at the end of the presentation. Good. Centrifugation is practically the one and only method in processing suspension cells for staining and analysis. And now, I believe all of you are very familiar with the centrifugation. And while it is widely used, it presents multiple challenges. So DA cell is positioned to minimize these challenges coming from the centrifugation of cells. In a conventional wash process, we typically use centrifuge and multi-channel pipette. A wash of two centrifugation steps usually takes a total 15 to 20 minutes by undergoing 6-step process. I mean, centrifuge and then flick, resuspend, centrifuge, flick, and resuspend. In the end, it may experience about 10 to 6% cell loss depending on the type of assay. In contrast, DA cell takes care of the entire wash process automatically in about 2 minutes. Precisely, it's about 2 minutes, 12 seconds without centrifuging cells. The only step required is to place a plate in a DA cell washer. Without centrifugation, cells do not experience any external force or stress. When it comes to cell retention, DA cell method has demonstrated equal or better retention. And according to internal and external data, actual cell loss observed on a DA cell plate is primarily due to disintegration of cells during the process, particularly fixation and permeabilization, and not from washing. I'll talk about it more in the later part of the presentation. So in the next slide, I show you a short movie clip of how DA cell washer runs. In the movie, we show a plate dispensed with 2 drops of green fluorescent polystyrene beads and then food coloring solution. And after about 20 minutes of rest, these all settle on a plate. Let me start here. Then a user places a plate in a washer and hits the start button. The plate goes in and the fluidics head comes down to a plate. In this movie, the case of a washer was lifted intentionally to show the movement of the head and plate inside. The head has 192 nozzles total, 2 nozzles per well, 1 for dispensing and 1 for aspiration. During 2 minutes of duration, it performs 7 times of dispensing and aspiration each. When the wash is complete, as you see, the plate comes out. In these pictures, you can see that the food coloring solution completely gone, while the green fluorescent beads are retained on the plate. You may wonder how come the ink was washed away while the beads were retained. Just to clarify, the beads are not attached to a plate. They are naturally and gently sitting on a plate by gravity without any adhesion. The next slide, I show that the retention of beads and cells on a plate is really not a magic, although I use the word magic. It's a simple science. We use gravity and laminar flow, very well-known forces in nature. The secret is that we use these two forces on a flat wall-less plate. That's the secret. At the end of a slide, at the bottom of the slide, you can see the working mechanism of cell wash by DA cell. Cells are mixed and settled on a plate by gravity. The settling takes about 20 minutes. Once the cells are settled, the action of dispensing and aspiration of both cells do not disturb them, leading to a cell loss. I suspect some of you may find it's difficult to believe that cells do not get agitated and sucked up during the aspiration. Well, I mean, in the end, we have a number of customers who have verified and use on a regular basis, and their data speaks for itself. So in understanding this theory, I mean, we kind of try to hypothesize and then understand. What happens is that during aspiration and dispensing and aspiration, a fluid forms a laminar flow and minimizes turbulence of a liquid by flowing over the cells settled on a surface. By the principle of laminar flow, the velocity of a fluid becomes zero at the surface of a plate. At the very bottom of a plate where cells are, cells do not feel the fast flow rate of a fluid. Over this micron level of a thin fluidic layer where cells are not disturbed, washing happens by diffusion rather than turbulence or flow of a liquid. This is how we can wash the cells without losing them. Interestingly, when we perform the same dispensing and aspiration in a conventional plate with a physical wall, the action of dispensing and aspiration causes turbulence, which lifts off cells from the bottom of a plate, leading to a cell loss. So at this point, some of you may wonder if the laminar flow provides, whether the laminar flow provides sufficient rinsing of cells sitting at the bottom of a plate, because it happens by only diffusion, right? So we have a lot of data to support, yes, it does. And then again, later in my presentation, I'll show you one very clear example demonstrating that the rinsing is sufficient. In the next slide, I explain key features of a DA cell washer and plate. The washer can process up to 96 samples in parallel on a 96-well DA cell plate. A plate can be used partially over 20 days by storing a plate at 4 Celsius degree. One well on a plate can hold up to 70 microliter, while 50 microliter is the preferred volume we recommend. We have an accessory that can increase the volume per well up to 300 microliter. This is the accessory used for direct reading of a plate by flow cytometer or lysing whole blood directly on a plate. One well on a plate can process even 10 million cells, starting from as few as a single cell. In a wash process, a dispenser dispenses about 6 microliter and aspirator about 6 microliter and repeats 5 to 7 times. In the bottom of a slide, the left picture shows a plate with a 50 microliter cell culture media. So this is how it would look with a 50 microliter on a plate. And the middle picture shows about 1 to 2 million cells settled on a plate surface. The next slide is quite important, and it shows what I have explained, how cells are rinsed by laminar flow and then how many cells can be handled. So you may wonder how one well of this a small one well of a DA plate can process as many as 10 million cells. To be honest, that we can handle 10 million cells per well was even a surprise to Curiox team. In order to examine the capacity of a single well, we designed and performed a series of staining assays with a different number of cells per well, from a few hundreds to 10 million. In this slide, I show you the data of cell densities from 2 million all the way to 10 million. In the test, we focus on two aspects of an assay: how effective the staining was and how effective the rinsing was. The effectiveness of staining and rinsing determined the quality of the data, which could be expressed as a stain index. The left graph, before I talk about the stain index, the left graph shows the number of cells recovered. As expected, whether it is live cells, staining, or CD45, we can see that the cell recovery remains almost perfect. The right graph shows the stain index for both live cells and CD45 positive cells. Stain index is from an empirical formula reflecting the distance between negative and positive mean values, as well as the standard deviation. What I like to highlight is that the stain index remains the same from 2 million to 10 million cells. If either staining or rinsing is not complete by this laminar flow washing and then addition of the reagents, then we expect to see that the stain index decrease because either mean signal intensities will deteriorate or standard deviation will increase. This result of identical stain index was a surprise to us, because we thought that 10 million cells was just way too many and way too much for our laminar-based wash. Well, in the end, we are very excited to find that it remains the same, suggesting that DA plate can handle even 10 million cells per well. Yeah. So the next slide. I also talked about this cell retention, and then we have investigated cell retention by drop array systematically by physical counting. One of the tests we performed is described in this slide. We prepared 1 million live PBMCs and dispensed onto a DA plate. Then the cells went through laminar wash process where we dispensed and aspirated about 60 microliter 5 to 7 times. In this particular process, we collected back the aspirated 60 microliter after each aspiration and observed under microscope, counting the cells physically. In the very first aspiration, as you can see in this table, we saw about 500 cells aspirated. It means that washed away. Then in the subsequent aspiration, we saw just 100 cells or so per aspiration. Such a pattern of a cell loss kind of makes sense. At the very first aspiration, we expect to lose more cells because there are more floating cells. And once these floating cells are gone, cell loss is much less because laminar flow does not disturb cells settled on a plate. In a real assay, we perform all the staining and washing of the cells with those cells settled on a plate. Only the very first wash of an assay, in the entire assay process, we may see those floating cells washed away, but the number is negligible. So with our method, we can minimize the cell loss throughout the entire assay, and that's how we improve the cell retention for any type of cell staining workflow. Regarding cell retention, I also want to share one of the data from a leading academic institute who wanted to investigate and compare the cell loss quantitatively and systematically. In the assay, they performed 3 different wash methods: DA cell, conventional tube, and cluster tube. Those 2 tube methods were optimized in-house by the institute. They also wanted to compare, interestingly, two different permeabilization and fixation reagents by two popular suppliers. Furthermore, they also had two different cell numbers, 1 million and 200,000. In the test, as you can see in those graphs on a slide, DSL method showed way better retention for both reagents and for both cell numbers. Interestingly, they see different cell retention for reagent 1 and 2 by the conventional methods, while DSL showed the same external retention for both reagents. In this slide, I also want to point out that drop rate shows about 10% cell loss for 1 million cells and around 30% cell loss for 200,000 cells. From our own internal tests, we actually speculate that the process of permeabilization and fixation naturally destroys cells about 10 to 30%. We know it because before or after permeabilization and fixation, we do not see such a dramatic cell loss. We only see such significant cell loss right at the wash right after the treatment, suggesting that actually the cell loss is most likely coming from the disintegration of the cells or damaging of the cells, not the wash. The next slide shows one example of immunostaining analyzed by fluorescent imaging by another customer. In this study, a customer was tracking endogenous protein expression in cells and compared those by conventional centrifugation and those by DA cell. Originally, the customer was interested in DA cell in order to retain about 50 to 100 cells per well, consistently and reliably. Interestingly, the customer also found out unexpectedly that cells handled by DA cells showed much stronger endogenous expression and better cell morphology. This case study is one of examples demonstrating the benefit of no centrifuge on cell health and viability. In the earlier presentation, I explained that a DA plate can hold up to 70 microliter. However, this volume of 70 microliter may not be enough to read a plate directly by those plate readers attached to a flow cytometer. Often, a plate is required to hold about 100 to 150 microliter per well to be read directly. So with an accessory, a DA plate can hold up to 300 microliter and therefore be read by a flow cytometer. The graph on the left shows the comparison between a conventional U-bottom plate and a DA plate, where gray bar is U-bottom plate and maroon color bar is a DA plate. As the graph shows, the reading from a DA plate is as good as reading from a U-bottom plate. And according to graph at the right, even when the cell numbers are as low as 10,000, the recovery of cells by direct reading are comparable between U-bottom and then DA plate. Overall, the workflow of a DA cell plate is practically identical to a conventional plate or tube. The sequence of steps, the duration of incubation, and the way it is read are same. The only difference is that a DA plate can complete the wash in 2 minutes, skipping the steps of a centrifugation, flicking, and resuspension. And it does it automatically and then consistently. So it eliminates those accidental loss of the cells and reduces the potential variation from a user to user. DA cell is extremely easy to operate and maintain. The washer is compact with the size of a 1 foot cube or 30 centimeter cube. It only needs to be hooked up with a fresh wash buffer bottle and then a waste bottle. Otherwise, it is just self-sufficient. The priming and cleaning are done with a 70% ethanol and DI water. The user interface is very simple and it is easy to operate, just a few buttons. A user needs to type in a number of washes and the volume of a drop on a plate, and that's it. and then just press a start button to start a fully automated wash process. Because the machine is so small, the washer can easily fit into a biosafety cabinet and facilitate the entire cell prep and process within the cabinet. So in this short presentation, I explained the benefit and working principles of a DSL washer. In summary, a DSL washer can complete one round of washing in 2 minutes with excellent cell retention and practically no stress on cells. A full automation of a washer helps improving consistency and reproducibility. And as I mentioned, we launched this product quite recently, and we are currently offering various purchasing options so more scientists can benefit from the product without this financial constraint. So I really thank for your time and attention, and I'm happy to answer any questions. So if you have any and you have not submitted yet, please do so now. It's not too late, and then I'll answer as best as I could.
Wonderful. Thank you, Dr. Kim. We do have a few questions that have come in. What prompted the invention of this washer? Isn't centrifugation good enough?
Right. So, I mean, I briefly mentioned those are three scientists who actually motivated us to develop. There are also other scientists, but primarily, we work with them, and then we found out that the handling of suspension cells was so much pain. Centrifugation, for example, requires a certain minimum number of cells to form a pellet, and then also the process is so stressful to the cells, and at the same time, it can be quite damaging for the staining too, because we have seen some of the data where the surface markers can kind of cross-contaminate during the pelleting process, right? And most of all, it causes good pain, and then there's a time to scientists to process this tedious, the centrifugation steps. So all these variations, stress on the cells, and time were the primary factors that we heard from actually our customers, scientists, and then identified that, oh, this is a need in the field, and motivation of the development.
Great, thank you. We have another question. Is there a maximum number of antibodies and stains that will fit into the small volume of the well?
Maximum amount of antibodies. We try to have the antibodies within about 50 microliter, because that's kind of the comfortable volume to handle on this wall-less plate. And so far, we haven't had any issue of staining using the antibodies. And I understand that sometimes antibody comes with a certain volume that's, for example, that we must use 120 microliter, 150 microliter. Actually, we can have the same concentration and then the staining even with just a 50 microliter. I mean, it's a bit long to explain here, but by simple liquid handling, we can achieve the same sort of the staining effect. So that would probably answer the question.
Yes, thank you. How can you pipette into the well if there's no walls?
Oh, yes. It's for that one. The best answer is seeing is believing. So if you look at our website, actually we have other products that we have this hydrophilic pattern, hydrophilic surface surrounded by a hydrophobic surface, and we hold as small as like a 3-5 microliter per well, and this 50 microliter is quite easy. And when we have this plate of a hydrophilic area surrounded by hydrophobic surface, the aqueous solution is really held by this hydrophilic surface by surface tension. And surface tension is surprisingly strong when the scale of the assay is small. So still 10 to 50 microliter, we managed to be controlled by surface tension, but not by gravity. So we could handle this volume of the reagents quite easily.
Good. Can I read directly from the plates?
Yes, I mean, I had one slide showing this direct reading from our DA plate. When we launched our product first, we didn't have that accessory, and then we received a lot of comments saying that, hey, reading directly from a DA plate would be so much easier to use. So we developed and came up with this accessory. The accessory is a basically plastic piece. It's a grid with the through holes. And then we can just drop it on our plate, wall-less plate, to transform our wall-less plate to walled plate. And after we just drop it, we can just add a much bigger volume of the sheath fluid, say 150 microliter, to read directly from this flow cytometer. And this grid, by the way, is reusable. Just we rinse it with water and then just keep using. So that's how we read the plate directly.
Good, thank you. How does cell loss compare between the centrifuge and the plate?
It's quite an interesting question. We have found that the cell loss of by centrifugation can be actually very minimal. We have seen a number of very talented hands-on scientists who can have an excellent cell retention. However, the challenge was that even with their hands, cell loss can really vary. Like by centrifugation, sometimes they have a 90% retention, sometimes a 70% retention. And when it goes to another scientist, another operator, the variation is even bigger. So I would say that centrifugation can have a very good cell retention, but the problem is that it's not reliable, it's not consistent. And drop array, it shows basically the best cell retention a centrifugation can achieve, and it does it consistently every single time to run. So we are not really making the cell retention much better than the best data of the centrifugation, but we can have this best data every single time. Yeah, that's probably what sums up the cell retention comparison between drop array and then centrifugation.
That's good. How do I scale my antibody concentrations if I have an existing SOP?
Yes, so we have worked with a number of accounts. I'll try to explain. So basically what we did is that, let's say you come with a 1x antibody cocktail of 150 microliter and you have to put it in a drop array plate, which can hold only 50 microliter. Then what we do is that we just have a pipette, we bring to our GA plate and then just mix up and down. There's always some residual wash buffer on a DA plate as well as a conventional plate. So with about 1/3 of this antibody cocktail solution, we pipette up and down to equilibrate the wash buffer. We dilute out the wash buffer. And then we remove the volume of this antibody cocktail, and then we bring a fresh antibody cocktail to add. By that time, the existing wash buffer is diluted away enough. We basically have a 1x concentration of this the antibody solution. And by that point, the volume doesn't matter because the staining reaction is driven by concentration, not by volume. It's by concentration because it's antibody antigen binding kinetics. So we have this good complete antibody solution, and we can have the same staining effect. So this is how we can achieve this the antibody staining even with this 1x concentration of a much bigger volume. And so far our data has demonstrated that yeah, it works, and we didn't see any difference.
Okay, with the accessory, can I load more than 50 microliters?
Yes. So again, coming back, this grid I explained, the reusable grid, we put on our plate, and then with the grid we can have up to 300 microliter of the reagent. So we used it for this whole blood lysis, where we have to have 1 part of the blood and 9 part of the or 10 part of the lysis buffer, for example. So yeah, more than 50 microliter, yes, we can handle with this grid.
Is it easy to clog the washer?
Oh, clogging. So some of the users we have met are familiar with a plate washer, conventional plate washer, and they show the concern of this clogging, with our washer. I want to highlight the main difference between conventional plate washer and our washer. Our washer, as I have explained, has syringes, 192 syringes assembled in a machine. So every single needle or nozzle has a isolated independent syringe plunger, and the plunger going up and down to push the liquid and aspirate. And you can easily imagine that soft materials like protein cells, there's no way you can clog the needle when you have this physical syringe going up and down, every single time. And our needle size is about 700 micron, so it's way too big for to be clogged. In contrast, in a conventional plate washer, it's just made of a reservoir with through holes, and there is the air pressure or big pressure coming to this reservoir to press down the liquid or aspirate the liquid. So by law of nature, if there's any clogging starting to happen in a one needle, then the resistance is higher, so the liquid will go to other needles, and then it aggravates the clogging of this accumulation within in that particular nozzle being clogged. So this is why in a conventional plate washer you may see clogging, but in a DA washer, because we have individual isolated plungers, you are not going to see this clogging.
Thank you. What information do you have to convince my boss that I need to buy one of these?
It's a very good question. The manpower saving, the time saving, we had one example. We had a 1.5 hour, this 2 incubation, 2 centrifuge wash step assay. We work with one of the customers. Their assay typically takes 1.5 hour. We saved it half hour to finish in 1 hour. So the productivity can increase. And if this doesn't work, saying that, oh, you gotta work out no matter overtime, I don't care, you must work. Then another benefit is that the consistency and reproducibility. So we minimize this the time to repeat the assay because we are not sure whether it was done properly and with some potential errors. So consistency is one good factor. And another good factor is this. I know that a lot of the listeners are coming from discovery. And some of the assays being developed in discovery team goes to this screening team, and then this to scale up the assay. And the automation is very critical at that point. And if you look at the centrifugation steps and drop array step, drop array is by far a lot more convenient to automate because it's already fully automated with a wash, right? It's a plug and play. So the return of the data and the assay is much faster from this screening group when the assay is being developed and executed by drop array. So those are the kind of justifications. And if those justification doesn't work, then don't work. Then another justification is that if we want to, we can save the reagents in quite significantly. So the cost saving is something that a lot of managers would like to hear, and so that's one of the benefits we can kind of try and then realize by drop array.
Good, thank you. We have a question about price. What is the cost of the instrument and what consumables are needed?
Right. Currently the list price of the washer is $49,800 USD, and then we offer early adoption discount, and then we also offer this installment plan where we kind of you don't need to, a customer doesn't need to pay upfront the entire amount. They can sort of pay off through the purchase of the consumables. And then the consumable, the plate, it's the lease price is about $48 in USD, But if it is purchased by like 100 plates at a time, the price receives about 40% discount to become about a little over $28.
Good. Another question, is it possible to use part of the plate in the first experiment and use the leftovers of the plate in a consequent experiment?
Yes, it's actually quite easy. I mean, we have a customer who has used over a period of like 3 weeks. It was not intended, but it happened to be, and the data was as good as it was at the first run. So the plate undergoes this the washing process by our washer. So every single well will receive this wash buffer. But let's say that I only use a half of the plate. The other half of the plate did not see any cells. It only saw the wash buffer, right? So we store the plate in a fridge, and then whenever it's ready to use again, we just take out the plate, we tap it to remove the whatever wash buffer in a well, and then just use those wells never seen cells before, never used for the assay. And yeah, this wash buffer only passivate the surface to prevent any cell adhesion. So it's only better for the assay, and there's no harm. And this is how we use a plate at a different time point.
Good. Can this technology be used for CyTOF assays?
Yes, actually, we have worked with a number of CyTOF users because, I mean, CyTOF is a great technology. It can really read, many more numbers of biomarkers at once. The challenge there is that the cell prep can be a little bit more tedious than the traditional color flow assay, and our washer can dramatically improve the workflow. And interestingly, the cell retention can be also improved significantly because the CyTOF workflow can be quite stressful to the cell. And then we demonstrated working with a number of CyTOF users that cell number can truly increase using our machine.
Thank you. Can the wash buffer be easily switched?
Yes, the switching wash buffer is not too difficult. We have this tube, and just we move the tube to another wash buffer, and then we hit a button, priming. It takes about 2 minutes plus, and then it will replace the existing wash buffer in a machine with this fresh wash buffer. So this is how we change. And for the automation, we can have this automation buffer change module attached to this our washer, and control the change of a wash buffer automatically by this scheduling software.
Good. Can this unit be placed for the shared usage at a core facility? What will be the challenge? Can the plate be placed in a full unit for washing only?
Yeah, so using this machine in our core shared facilities, I mean, it can be done. Actually, we discuss and then work with a few like core labs. So, for example, the unit can be stationed in a like a cart, and then it can move. Whoever need to use it in their own lab. That's one way to use the machine for the core facilities, and then shared by different groups. So as far as they follow the maintenance and then do properly, it can be shared by different groups at a core facilities, provided that the cells are prepared by individual labs. And if cells are prepared in a core facilities lab, then of course, I mean, it's easy. They just bring the cells and things and just prepare the cells in the core lab using a drop or a cell washer.
Who is the ideal user of this washer?
Actually, I would say that anyone and everyone using running flow assays will definitely benefit from our washer. And then among those flow users who run a number of plates, so the time spent on this plate prep cell prep is quite significant. We can definitely help them. And also those who runs a small number of cells per well, we can definitely help them. And then those who experience, significant cell loss and who are concerned about this biased relative population of the cells due to the non-uniform loss of the cells, we can help them. And lastly, those who see this the cells affected by centrifugation in terms of the expressions or pathway studies, DA cell can eliminate those stresses. So I think basically most of the flow users do experience one of these challenges I mentioned, and they can benefit from our technology and product.
Great, thank you. Well, that looks to be all the questions that we have. So thank you so much, Dr. Kim, for presenting the technology. And please visit our website at curiox.com. And please continue to ask questions. We can always email you with an answer. Thank you so much, everyone.
Thank you. Thank you, everyone.